we might get started with the next session. So our next speaker is Edison Durigon from, I can't speak Portuguese, so I'm not going to pronounce that, but from Sao Paulo in Brazil. He's Professor of Virology and Microbiology and has worked as a guest researcher for the CDC in Atlanta. So he's going to be talking to us about wildlife monitoring in Brazil.
Thank you very much. I really appreciate to be here. Thanks for inviting me to come for this nice talk about coronavirus and the opportunity to talk about the Brazilian network surveillance of coronavirus in bats, birds, and the other forest reservoirs in context of One Health. Just to position everybody where we are, it's Brazil. It's a large country in South America. We speak Portuguese, no Spanish. It's the only country that speaks Portuguese in South America is Brazil. And this is University of São Paulo. I'm professor at University of São Paulo in microbiology department. And this is our department. This is our building. This is microbiology building. And this is São Paulo City, and this is the whole campus that you have at the university. It's a huge university. São Paulo is a big city, it's around 12 million people who's living there. It's like London, New York, or something like that. And we have 2 BSL-3 facilities, and we have a field equipment to working and looking for reservoirs or wild birds and wild bats. I will talk only about our work in coronavirus in bats, but this network, we do a large number of viruses, because once you go there and capture these animals, you should do everything that we can. Now we are facing the avian influenza virus in Brazil. We get big problem with migratory birds that come from the North America to South America every year. And it's around 5 billion birds that migrate every year in October, and then they go back to North America in May, it's right now. And they bring virus and bat virus from South America to North America. Now we started last week, we have our first 8 cases of H5N1 in Brazil. It's a big problem because Brazil it's a huge in terms of exportation, and they have all these chicken farmers and everything. And the government is like crazy right now. We also have this project that is a federal project that when the pandemic started in 2020, it's like March, the government reunited all the Brazilian Virology Society to create what you call virus network, or in Portuguese, rede vírus. That is with mainly and support and financial support by Minister of Science and Technology. The idea of this is to monitor not only SARS-CoV-2 in the human population, but also to monitoring the coronavirus in wild animals. And I was responsible with Dr. Clarice Arnes and Helena Ferreira to coordinate this project they call Previr. We try to predict the virus before they starting. And we select several hotspots in Brazil when you have a very conserved areas from different biomes, and the groups that usually work to collect samples in the field, because it's hard. You have people that go to the field, capture bats, capture birds, and then bring to the lab and do the same work. We try to create a mix of a group that work in the field and the group that work in the laboratory, for we can get some good research. Then we look for, now we look more for bats, because the coronavirus is emerging for bats, but we also look at different reservoirs. we create an applicative, it's app, to get all the data collection in a big computer that we have in São Paulo, Campinas. And then we have the people in the field collect swabs from blood, cloacal or oral, and the rectal swabs. And also we collect the blood and the tissue samples when it is possible. The majority of these bats, we collect samples and relieve there. Okay? Some bats will get some problem, or they die, or we capture, and then we collect the tissues. But the idea is do not disturb this environment. And then these samples come from liquid nitrogen to the laboratory. We have 4 reference centers I show for you. And then we screen by PCR. For coronavirus, we use the primer from the polymerase primer, is Xu et al. 2011. It's a protocol that we use. We work with Predict before in the US program, and then we use the same primers for that. Then all the positive samples, we sequence by Sanger, and then for NGS, and do the phylogenetic analysis, and the positive samples we try to cultivate in cell cultures. That was another big challenge with this virus for us. Okay. We have 4 biomas that we're looking for. It's Amazonas area, Atlantic Forest, Cerrado, and wetlands in Pantanal. That is some numbers that when we start the field activity is in November 20. 1 year after, we already have more than 2,000 bat samples collected and more than 2,500 bird samples collected from the 4 biomas that we studied there. That's for we get a better idea. I talk about, we work in the Amazonas forest. That is a very pristine area. We also work in the center of Brazil that we call Pantanal. It's a huge wetland area that is has a very dense wild animals population. Then we have the South Brazil and Southeast Brazil, that our Atlantic rainforest. Our Atlantic forest is very conservative, it's very pristine area. In some places they are very degraded too. But this is a large forest that's come to the northeast to the south of Brazil. And then we collect in different points of the country. And here in the extreme south is our big hotspot for birds, because when they migration from the North America, they usually come from this area to feeding, etc. That's the way that we work. We have these 5 field teams in 4 different biomas. That is the same that I just catching. And one way for we work is using these mist nets. It's a horizontal mist net that you collect the samples. This is one picture from the Pantanal, the wetlands. This is our symbol, our bird symbols is Tuyuyu, one of the most beautiful flying that I saw in my life. That is, we put the nets here to capture these birds during the night. And also we search in the shelters, use a kind of a net trap in the houses that you go in the ceiling, under the bridge. They have a lot of bridge there that is abandoned like that, or it's in the middle of nothing. Or even in the platforms in the river, that the bats stay down this. And we go there and capture with these traps. That is only for you guys have some idea as to how many bats we have there in all these ceilings. When you capture, we do this field work. We have a team that is prepared for that, and then we collect swabs from oral, anal, and we also collect blood for those serology studies. I do not have any serology data yet because we didn't set up the ELISA for these kinds of blood. But we swear that soon we get some. We still try. And when we had tissues, we use this kind of field glove box where we can work there, and then we can take lung, kidney, intestine, or other tissues, and put in the vials and send to the laboratory in liquid nitrogen. And that is the most common species that you have from the bats there. Some are hematophagous, like the Mosus rotundus. And that is the total work that we are doing during 2021 and 2022. We also have some dates from this year because we're still very activity. But we are more focused this year in avian influenza than in coronavirus in bats. But the group is still working there. We have in the total more than 6,000 samples collected from the animals, 6,000 animals. And the samples we have more than 29,000 collected in all these different places in Brazil. Okay? Only the bats we have 3,580 samples from both years, which more we have. And we also have a passive survey. A passive surveillance, we have a big institute in São Paulo, that is Pasteur Institute, that has nothing with the Pasteur Institute in France. They have the name, because when they building this from our government, they build in the same way for the Pasteur Institute to looking for rabies virus. Right? That is focusing in rabies, like Dr. Enio is responsible for these samples. And they have several local organs that when they have some bats that get dead in the backyard from the people, and people call there and they collect the bats and bring it to Pasteur Institute in São Paulo. Also, we have a branch in a countryside of São Paulo, in Ribeirão Preto. And then this is go to Dr. Enio that do rabies, and all negatives for rabies, they send to us. Okay? And then we do a passive surveillance. We have, only for you get some idea, this is for the density of the number of samples that they draining for this institute. Where the dark ones is more than 5,000 samples. That is many people. This is like a tradition. They collected this and sent to Pasteur Institute. That's worked very well for us. Okay. Now that's the idea of the preview. It's have all the collection of the biodiversity data. That is working very, very well. And we are facing some challenges because it's not easy. You put different groups in different states, because you imagine the size of Brazil. Brazil is the size of the United States. It's almost the size of Europe. And each one in different places doing the same protocols. Okay? Different biomes. The work in the field is not easy. And then we try to make a very detailed protocol, but get everybody doing the same is not so easy. Okay? But we tried our best. It's doing well. We have a nice form that they fill up and put in a computer. They can use it the cell phone. But you imagine, they collect the bats in the night. They take to the net. They go there and collect swabs, collect blood. And then also they need to put all the swabs and bloods in the vials, put in the liquid nitrogen, and they also need to fill the form. It's crazy. It's really crazy. But they are doing the best they can, and we get a nice results with this. Okay? All this data collection is very important. Okay? And then because we link the sampling with the web app and the laboratory analysis. and then we get better results in our. So also we digitally liberating ecological knowledge from local publication. That is one of our goals in this project, and give all this information for different groups in Brazil, that they know what we have, how many species we collect, what the virus is going on, and let's make very, very nice database and collect data. We also have our post office, we call Correios. It's a federal agency that they do exactly the post office doing. They go to the reference labs, take the liquid nitrogen, send it to the people in the field. When the people, they spend, for example, 3 or 4 days collecting blood swabs from the beds, and then they come back there and collect these samples and bring it to the reference labs. This is make more safe our transport, and we cannot lose it like cold chain and everything, because the worst scenario for this kind of work, you go, for example, in the Amazonas area, you collect 300 samples from bats, and then when they come to the lab, they are warm, they are not good to work, etc., etc., etc. Then we take care of this. This federal agency is working very well in Brazil, and then they have these 4 reference centers for do the laboratory work. Okay, basically, we get the animals, get the samples, and then we develop some PCRs for doing the selection. We are now trying to get some cell lines from bats, and Dr. Clarice Arnes is in Campinas. She's our expertise in cell culture, and she has 3 or 4 bat cell lines working there, and then she gets some bats that we are common in Brazil, and try to make a primary cell line and try to immortalize that. I don't know what we have. And we also try to improve our NGS protocol, because we are able to collect the data, sampling, do the PCR, we get a positive, we are able to sequence from the polymerase gene. It's a short piece, but you get a good sequence. We know if it's alpha coronavirus or beta coronavirus. We can make a tree, but we are not able to get the whole sequence, because we get the samples from the bats that is flying. They do not have too much virus there. The CTs, 90% of the time is over 30. That's difficult to do the NGS there. We are facing a large problem with our NGS protocols. Okay, we try to do some enrichment technologies prior to sequence it, to see if we can get the full sequence of this virus. The idea, we have to grow this virus in cell line, and then we can sequence easily, or get some sick bats, then we get a lot of virus, but we don't have both. So we can't grow yet, and we don't have this kind of bats. And we still have this gap in our studies, but now we have a huge work to gather some companies like Illumina to try to develop these probe target sequences for a pan-coronavirus, because it's easy. You sequence SARS-CoV-2. You have a lot of kits there. You can just throw there and get the whole sequence. It's nice. And when the patient is sick, you get a lot of viruses and make it easy. But when you have a coronavirus that's completely new, you never have how you do that. Okay? And another one is try to depletion of mitochondrial DNA using Cas9 system by CRISPR-Cas, and see if you get some bad results. We also working a lot with the bioinformatics group. That's not easy. We have a special group who's doing this platform for us to try to make easier our data. We have bunch of data from the reservoir, but not from the virus. That is make us very, very difficult. But the bioinformatics is working very well, and maybe soon we get some better results. Now, let's see what we get in these 2 years. We have some collection samples that from south of Brazil. We use some undisturbed forest remain area in Iguaçu Foz. It's a nice place there. And that still is a pristine area. We have some fragmented forest areas in the center of the states of Paraná, that is a south state, and the urban areas in São Paulo and in northeast of São Paulo too. That we looking for these 3 different situations. It's a pristine area, a degraded area, and the urban area. We still have some difference. This work always we have several positive samples for bat coronavirus. We search more than 1,800 samples. We get 3.1% of positive. Yes, 26 is positive. We get 10 bat species in this area. 11, which is a coronavirus genotypes. 3 is a potential new coronavirus. We have 24 alpha coronavirus and only 2 beta coronavirus. We found much more alpha coronavirus in our bats than beta coronavirus. But maybe it's the area we're looking for. We have a huge diversity of this beta coronavirus and the alpha coronavirus. And the beta coronavirus that we get, some get similar to MERS. It came in the same clade. Okay, it's true that we collect. Okay, I go. And they show that we have this large. Brazil has a large diversity. That's another data that we have from alpha coronavirus detection. We get from almost 1,000 samples. We have 44 samples positive for coronavirus. That is the bat species in 8 bat species. One is hematophago. It's a Diphila eucadata. And this is the sites we have in the northeast, south of Brazil, and southeast to detect all this sample. That is the way that we detect. We get here in this study 3 new bat coronavirus hosted in the remain forest in the tropical area. That is the sequence that we did. I will not go in detail because it's too small for we understand. But we have at least 4 subgenera circulate in Brazil. Okay, that is the subgenera that we have collected there. It's one thing that is very interesting because we have different location with the same species of bats, and we find the same virus. That we start to think if it's possible this bat migrate from one site to another. We don't have any information about bat migration. That is a big gap in our situation. And then we have some in tissues from these hematophagous bats. I go very fast. We did some sequences in the S gene, that is the spike. And in summary, we have collected bats in the whole country, and we get almost 12% of the positivity in all these bats. One interesting data is we get more coronavirus in area that have more percentage of cover by forest than in degraded area. It's something that helps. We also find SARS-CoV-2 in a Nasua, it's a coati. This is it's a kind of a raccoon that is very common in Brazil. We found a Zeta variant virus in this raccoon. We have 2 positives. We test for 2 different areas of the genome. And now what we are doing is do virus isolation to try to get some better results, develop a new cell lines. And what we pray that we need good conditions to work in Brazil with all these governments and etc. That is the whole group that we have. That is Helena Lage and the Clarice group. That's my group in the laboratory. And that is the guys that work in the field. That is the most difficult work. That is the financial support that you have. We cannot complain about the money. And that is we always pray in Brazil. That is, okay, see. Thank you very much.
Have we got questions for Edison? Oh, no, I have questions.
Thank you for the nice presentation. What percentage were you able to, you said sequencing was difficult because CT values were too high, but what percentage of your positives were you able to sequence? And that probably differs for the nasopharyngeal swabs compared to the rectal swabs. Do you know how that differs?
We have, for NGS sequence, we have not. We only have the sequence from the polymerase area. We have around 12% of the positive for that. But
and also from the rectal swabs? Or?
Can you move back to the microphone?
Oh, sorry.
The positives, you said 12% positives of your positives. 12% you were able to sequence from your positive samples?
It's already sequenced.
Okay. And how does that differ for the rectal swabs compared to the nasopharyngeal swabs?
Sorry for my Brazilian English.
The rectal swabs versus the nasal.
I assume that the rectal swabs.
Yes. We get more positive in rectal swabs than in oral. Okay, but what do we do? We collect samples from swab for oral and the rectal and put in the same tube for screening. And then we collect from rectal and swabs in separate tube. Okay, because if disease gets positive, we go back and then we select. What we find, we get more positive in the rectal than oral swabs. Okay.
Okay, thank you.
Got another question up the back.
Thank you so much. This is such critically important work. As you know, this is a huge gap that we have in animal health, is the lack of databases that are dedicated to accumulating genetic information for a lot of our pathogens. Yeah. So these are very expensive to maintain, and this is critically important work. My question is, out of curiosity, as you know, I know you're familiar, I'm sure, with GIS-AID, right, which was actually started out by veterinarian under the OFFLU umbrella with FAO and the OIE that started out trying to do the same thing with influenza viruses, and it eventually, merged and was taken over in Germany, and they started to focus on coronaviruses before the pandemic, and that ended up being a key tool to understand, the emergence of many coronaviruses. So out of curiosity, is your center, your surveillance program that is focused on coronaviruses in wildlife, have you been connected with this GIS-AID? Is any effort to connect the two together to enhance the value of these databases in terms of perhaps predicting in the future what is likely to emerge from this important family of viruses? Thank you.
Okay. Yes, the answer is yes. We are connecting. We have a group on this virus network that called Corona-omic, that is connected straight with the GS group, more for SARS-CoV-2 than for others, but they are trying to help us in this kind of analysis. Yes.
So I think we'll have to leave questions till coffee time when you can find Edison afterwards. We'll move on to our next speaker. Thank you.
Thank you very much. Thank you.
Thomas, do you want to come up? Can we get... So our next talk is staying in the Americas, but different part of it. So we've got Thomas DeLiberto from the US government organization, who's going to talk to us more further about SARS in deer.
Thank you. Well, thank you, and good morning, everyone. I have the honor and the privilege of representing my co-authors from the U.S. Department of Agriculture, University of Missouri, and the Centers for Disease Control and Prevention in providing you this update on cervids in North America. So back in 2021, the U.S. government passed the American Rescue Plan Act. And this act was passed to help the country recover from the COVID-19 pandemic. And as part of that legislation, USDA received $300 million to conduct surveillance and monitoring in animals thought to be susceptible to the SARS-CoV-2 virus. So in the next few minutes, I'll be discussing some of the activities specifically related to cervids, wild cervids, that we've been conducting as part of this American Rescue Plan Act program. So as we've all been talking about the last couple of days, since the beginning of the pandemic, there's been a number of species that have been documented to be susceptible to SARS-CoV-2 infection, either naturally or through experimental infection studies. With regard to North American wildlife, we now know of 26 species ranging from herbivores to carnivores and rodents to cervids that have been shown to be susceptible to the virus. Of those, SARS-CoV-2 has only been documented, the virus has only been documented in five of those species in the wild. There was, we mentioned earlier this morning, an American mink that was found positive in 2020 associated with a positive mink farm. There was a Virginia opossum that was found positive associated with a household of humans that were concurrently positive for the virus. We've found 4 brown rats in New York City positive during 2021. And of course, we found white-tailed deer and mule deer that have been positive. Those are the 5 species that we found positive in North America, evidence of virus in free-ranging populations. Now, with respect to free-ranging cervids, particularly in the United States, we've been looking for SARS-CoV-2 in a range of species since 2021. To date, though, we've only found evidence of the virus by PCR in white-tailed deer and mule deer, although we still have a lot of testing to do in the other species. Most of the other species we just recently started doing surveillance in this past fall. We're also collecting blood samples from these species. We've got positives only in white-tailed deer, primarily in white-tailed deer, to a lesser degree mule deer, and again, lots of testing yet to go in the other species. So given that most of the testing and results we have in the United States and in North America is for white-tailed deer, I'm going to focus the rest of the discussion on that species. So we've mentioned earlier that in 2021 we published our first paper showing that prior to 2020 we could not find evidence in archived blood samples of SARS-CoV-2 from wild deer, blood samples taken from wild deer. And then once the virus was introduced into North America in the human population, we very quickly saw the presence of antibodies in the deer, and by 2021, we saw a dramatic spike in antibodies in wild deer, white-tailed deer. So that study was quickly followed by a series of experimental infection studies by USDA and some other university partners. And all of these studies quickly documented the susceptibility of white-tailed deer to SARS-CoV-2. They established shedding patterns for the virus in the species, primarily through oral and nasal routes. They documented efficient deer-to-deer transmission in captivity in BSL-3 containment. And all of the studies demonstrated that there were no overt clinical signs presented by infected animals. So now those experimental infection studies were then followed up by the first set of field surveillance studies. And these studies demonstrated that SARS-CoV-2 virus was present in wild deer populations, white-tailed deer again, and those animals were shedding virus primarily through the oral and nasal routes. And then they also documented for the first time local deer-to-deer transmission in the wild. So given those early research studies, both in experimental containment, BSL containment, and in the field, USDA partnered with our state, federal, and tribal partners in 2021 to initiate national white-tailed deer surveillance in the US. Then later, this past year, as I mentioned, we started surveillance in mule deer and other cervids. Again, from here on out, I'm just going to focus on the white-tailed deer stuff. That's where we have most of our data. Now, the objectives of these projects are to determine if SARS-CoV-2 is present and circulating in cervid populations, and if there are significant state and regional differences in circulation and prevalence among those sampled populations. And really, the ultimate goal is to determine what are the transmission pathways and whether we have or can develop tools to interrupt those spillover transmissions from humans to white-tailed deer. So just turning to some of our initial surveillance data, if we look at the first year of surveillance, that again is November of 21 through April of 22. We see that we tested a little over 9,000 nasal swabs, or nasal and oral swabs, across the range of white-tailed deer and found a prevalence of about 12% positive. Now, in this map, the green states are the range of white-tailed deer. So those are the states that white-tailed deer are found in. The size of the circle represents our sample size, our relative sample size. And the shading of the circles represents the prevalence within the state that the samples were collected. So one of the first things you'll notice here is that the darker circles tend to be more to the east and to the north in the US, and that just so happens to be where most of the highest densities of not only white-tailed deer, but humans are located as well. If we look at some of the initial serological results from that first year, we found a mean prevalence of about 31%, again with the highest levels in the north and the east of the country. So of those 9,091 PCR positive samples, we found, 9,091 nasal oral swabs, we found about 1,000 samples, or 1,116, that were RT-PCR positive. We then looked to sequence those samples. Of those, 392, sorry, this is a typo, 392 of them, we obtained samples, whole genome sequences for, and of those 392, we were able to get 282 sequences where we had greater than 95% coverage of the genome. And so we were going to focus our our discussion from here on out on those 282 samples. Of those 282 samples, we had 34 Pango lineages from Alpha, Gamma, Delta, and Omicron variants. And while we got a great diversity of viruses, we tended to see similarity in lineages represented within the populations within specific regions. So the Northeast region of the country, the Midwest, and the Southeast. Then if we look over here at the top, if we look at the top, this is a graph of depicting the frequency of the variants of concern in the human population by time. And the dotted line here represents our sampling period from November through April of 2022, November 21 through April of 2022. You'll notice that we found that Alpha and Gamma variant in the beginning of that sample period still circulating in white-tailed deer well after those variants had disappeared from the human population. Most of the samples were Delta variant, which coincided with the peak in Delta variant in the human population. And then towards the end of that sampling period, we start to see a few samples of Omicron being detected. So next we looked at those 282 deer viruses and we compared them to 14 million sequences in the public databases and identified human precursor viruses that were similar to our white-tailed deer viruses. From there, we grouped them into 3 transmission events. First, human to white-tailed deer, consisting of at least 1 human precursor virus, followed by a subsequent deer virus. Human to white-tailed deer to white-tailed deer. So this consisted of a human precursor virus, followed by at least 2 deer with similar viruses. And then human to white-tailed deer back to human transmission events. Those consisted of 1 human precursor virus, followed by multiple similar deer viruses, followed by at least 1 virus back in the human population. Once that was completed, we were able to identify 109 transmission events, the majority of which were human to white-tailed deer, followed by human to white-tailed deer to white-tailed deer, and then finally human to white-tailed deer back to human. Of these, all but 11 were Delta variant. The 11 were Alpha variants, and we didn't identify any transmission events that included Gamma or Omicron, at least in that first year of surveillance. So of the 109 spillover events, we saw 833 amino acid substitutions, of which 112 were repeated at least twice, and 58 of those were under positive selection. So the columns here represent the 58 that were under positive selection, amino acid substitutions that were under positive selections. The top graph here represents the number of times we saw an amino acid substitution, identified in red here in these boxes. The x-axis represents the number of the 109 spillover events, and the horizontal line separates the specific lineages identified in the variants of concern. And then finally on the bottom are the proteins in which those amino acid substitutions occurred. There's a lot happening here. Sorry about that. But one of the important things to take away from this graph is that we had 9 different repeated amino acid sequences that were under positive selection in the S protein, the spike protein. I've put them in red here on the right. And you'll notice that none of those occurred in the receptor binding domain, suggesting that the virus was well adapted to the ACE2 receptors in white-tailed deer when it spilled over into the populations from humans. So in summary, we had 9,091 respiratory samples that we collected and tested for for SARS-CoV-2 in free-ranging deer from 29 states in the US. From November of 21 through April of 22, we obtained 392 sequences and identified 34 pango lineages in the alpha, gamma, delta, and Omicron variants of concern. Evolutionary analysis showed that these white-tailed deer viruses originated from at least 109 independent spillover events, which resulted in 39 cases of subsequent local deer-to-deer transmission. Some of these transition events did span several hundred kilometers in distance. And we did identify at least 3 cases of potential spill back from deer to humans. And I emphasize potential because what we don't have here, this is all based on genetic data, and we're not presenting epidemiological data here. And that's critical, as we heard earlier, I think yesterday, to have that information. Viruses repeatedly adapted to white-tailed deer with recurring amino acid substitutions across the spike and other proteins, but not the receptor binding domain. Overall, our findings suggest that multiple SARS-CoV-2 lineages were introduced, became enzootic, and co-circulated in white-tailed deer during 2021 through 2022. Now, I'll just give you a brief slide on some of the preliminary results from this last year of surveillance. We now begin to see a significant drop in the prevalence of not only PCR positive, but also in our seroprevalence from year 1 to year 2. And we continue to see circulation of the Delta variant. However, the Omicron variant appears to be more prevalent in our sample in this second year. So this suggests that we still have deer-to-deer transmission, and is evidenced by circulation of Delta variant well after Delta variant has been replaced in the human population. And we continue to see spillover of virus from humans to deer based on the identification of Omicron variants in our samples in the second year. So thank you all, and I just throw up this to acknowledge that just the tremendous work by a lot of people that were able to do this large surveillance effort and analysis. Thank you. Right on time?
So we probably have time for one question for Thomas. So Wim.
Thank you for this nice presentation. From your sequences in your different transmission scenarios, did you also try to make an assessment of the reproduction ratios of the virus? Because based on your changes in the sequences, you could make an assessment. There are programs to do that, to make an assessment of the reproduction ratios of the virus.
Yes, so what we did in another analysis we conducted, we did show that, which is actually in preprint, we did show that we saw more rapid evolution of these viruses in white-tailed deer than we see in humans. And I guess that's not surprising, since when you get a virus that jumps from a host species to a new species, you tend to see rapid evolution of those viruses. So, so we did see some rapid evolution of those viruses. But again, most of that evolution was in the internal proteins, right? The internal genes, not in the spike protein.
Yeah, I'm asking because what you would like to know whether the virus will persist in the deer. So if the reproduction ratio stays high, you could have a persistence of the virus in the deer, and that then could be a reservoir for the virus.
Yeah, R naught was above 1, we did that analysis in one state, and R naught was above 1 in Ohio, which is the state.
Yeah, and I know there were assessments of the R not, but that was mainly based on the longitudinal testing of the animals. But it would also be interesting to look at the sequences and use that information to make another assessment of the R not.
Yeah.
And see if it's the same or maybe higher.
Yep, that's a good point.
Okay.
Yep.
Thank you.
Yes, thank you.
Thank you, Thomas.
Thank you.
So I think the next speaker is actually me. So this is on a poster out. Oh, sorry, Des. This is on a poster outside, so you can go and have a look at it in a form that you can actually see if you wish to. But I'll just whiz through it briefly. So we were monitoring the UK wildlife that isn't deer. And at the species range selection was, at the time the grant was funded, the ones that were considered to be the highest risk because we had field or experimental data for them. So it's basically mustelids, cricketted rodents, and we have 2 species of horseshoe bats in the UK. And we were doing swab, or whatever we could get hold of, because some of these animals came from roadkill post-mortem programs. We had access to cadavers. Lots of them were from different field collection, long-running wildlife surveys. Lots of these were collected by volunteers, little local wildlife groups who were sending stuff in to us. Some of them very enthusiastically. The bat people were very enthusiastic about sending us poo. So the long and the short of it is we didn't find SARS coronavirus in any of them, using an e-gene qPCR and a pan-coronavirus PCR, which is a good thing. And we sampled quite a lot of animals in the end. We did find other coronaviruses, so any positives that we got, we deep sequenced, and happy to talk about the methods we used for those, because we recovered full genome sequences for a sarbecoviruses in lesser horseshoe bats. And close to 40% of the fecal samples from those animals were positive, so that's PCR positive, which is really, really high, which probably indicates a chronic infection in those animals. And that sarbecovirus has been detected by other groups and it appears to be quite widespread across the European populations. We didn't find it at all in the greater horseshoe bats, which, even though they share roost sites, we were quite surprised about, and we should have found something in the greater horseshoe bats. So I'm not entirely sure what's going on there, but I'm quite confident we didn't find it. The other virus we found was an alpha coronavirus of stoats, and these were, I was quite astonished that we got positives off these because they were rectal swabs sent to us from Orkney, which is the other end of the UK, and took days to reach us. And they were really poor quality samples we sent off for sequencing. The thing that made the biggest difference in the sequencing recovery was actually the bioinformatics methods we applied to the data afterwards. So Tom Hiron, who's part of Lucy Davison's project, is a very good bioinformatician, and we followed the methods he was using, which is a program called Corona Spades, which is used for metagenomics, but it has quite a clever mathematical trick in it that enhances the recovery rate of sequence we get out of it, because it basically puts them in the right order for the right proteins. And that made a massive difference to our sequence recovery. So we have a novel alpha coronavirus of stoats. It is quite closely related to the known mink and ferret ones, which cause diarrhea. We have not a clue about what these viruses actually do in the host species, their seasonality, whether they cause disease, whether they have an impact on them. And stoats are a really common species across Europe. So when I actually went and looked, we have astonishingly little data or information even on species that are really common. So we do have a massive gap and a hole in our basic knowledge of what's going on with these species. So that's really it, really. So the data's published and up on bioRxiv, and if anyone wants to have a look at it or get in touch, feel free.
Okay, thanks, Rachel. We've got time for one quick question.
Or not. That's fine.
I've got a question.
Go ahead.
So where next, really? What are you planning on doing next in terms of
So I
more
think
surveillance?
one of the things that's become really clear reading recently and looking at the work you guys have all been presenting at this conference is that the species susceptibility is very dependent on the strain of coronavirus. And I nearly crapped myself when I looked at Nicola's deer results, because all the other deer in Europe are negative. But because this is going to keep changing in people, that species tropism is going to keep changing, and the results we have at the moment, it's not going to stay static. So we do need to keep going with this and keep monitoring. It's going to be harder because there's a lot less interest now that the human pandemic is regarded as over. But we have a real risk with data like Thomas just presented that this is going to keep circulating in older variants that have vanished out of the human population. They will come back to haunt us, perhaps in new pandemic forms and perhaps not where we're expecting it from. So what we know now is not static, and we need to keep going.
Very good. Okay, thanks very much, Rachel. Our next speaker is Andreia Cruz.
Good morning. First, I want to thank you the opportunity to present our work on coronavirus in European hedgehogs from Portugal. The zoonotic emergence of SARS-CoV and MERS-CoV has raised concerns about the potential for coronavirus to spill over from wildlife species to humans. Because of the high number of bat coronavirus newly described, it was speculated that other insectivorous mammals could also harbor coronaviruses. In 2013, a novel beta coronavirus was discovered in European hedgehogs in Germany, raising concerns about the synanthropic circulation of potentially zoonotic coronavirus. This study aims to identify and characterize the diversity of coronavirus in European hedgehogs from northern Portugal. For this, fecal samples of 48 hedgehogs were collected in northern Portugal. Broad-spectrum pan-coronavirus nested RT-PCR was performed targeting the highly conserved gene of RNA-dependent RNA polymerase. Amplicons with the expected size were sequenced by Sanger sequencing, and the identified sequences were submitted to the gene bank. So we found that 33% of the hedgehogs tested positive for coronavirus, 40 sequences grouped phylogenetically within beta coronavirus belonging to hedgehogs, while 2 sequences correspond to alpha coronavirus, similar to bat coronavirus. Similar to other studies carried out in Europe, the prevalence of coronavirus in hedgehogs is high. However, to our knowledge, this is the first time that an alpha coronavirus has been identified in hedgehogs. In the near future, we will increase the sample size and characterize the identified sequences on the whole genome level. Thanks for your attention.
Okay, thanks very much. Do we have any questions? Right, I'll ask one. So have you got any plans, or have you done any serological sampling from hedgehogs?
Sorry?
Any serological sampling?
No. It's just fecal samples.
Just fecal sampling. Are there any plans to do any of that?
For not now. Not for now.
Okay, fair enough. All right, thanks very much.
Thank you.
So our next speaker, I hope I'm not going to mangle your name too badly here, is Leira Fernandez-Bastit.
Good morning, all. I am Leira Fernandez-Bastit, a PhD student at IRTA-CReSA, and I am working at the coronavirus group, studying the susceptibility of different animal species to SARS-CoV-2 infection. and I am going to present our study in which we are investigating the role of different animal species of wildlife in the COVID-19 pandemic. As it has already been explained in the previous talks, SARS-CoV-2 not only infects humans but also different animal species under natural and experimental conditions. Naturally, SARS-CoV-2 has infected 26 different animal species in 36 countries, including domestic and wildlife in captivity and in a free-range environment. SARS-CoV-2 has infected white-tailed deer in United States and in Canada. Also, it has infected a black-tailed marmoset in Brazil, and also different mustelid species, including one Eurasian river otter in Spain and feral minks in United States and in Spain. Although free-range animal species are not as close to humans as domesticated or captive animals, it is direct or indirect contact with humans may be considered a risk of infection for this group of animals, as well as their contact with other susceptible animal species, such as feral cats or, for example, American minks from farms. Considering that the spread of SARS coronavirus 2 in the wildlife is difficult to control, it is important to control the presence of the virus in this group of animals. For this reason, we start to monitor SARS-CoV-2 infection in different groups of wildlife animal species from Catalonia to assess their potential role in the epidemiology of the COVID-19 pandemic. On the one hand, we sampled murine species from urban areas from Barcelona, since it is a group of animals that demonstrate to have susceptibility to some variants of SARS coronavirus 2, although it is a group that is not susceptible to the ancestral variant. We include black rats from large parts in Barcelona. We include common rats from that were captured for the census in the sewer, and also we include common mice from buildings of Barcelona. We also include non-urban wildlife species, including a group of mustelids that we include American minks, Eurasian otters, beech martins, and European badgers. And also we included common genets, red fox, and coypus. Coypus is a rodent species that is considered an invasive species here in Europe. Also, the American mink is considered an invasive species, so these American mink and coypus are captured for control, for population control reasons, and the other animals were captured for other reasons, such as vehicle collisions or damage. We collect oral and rectal swabs and lung tissue from this species, and oral swabs and lung tissues from this group. We performed the detection of SARS coronavirus 2 RNA by RT-qPCR, and we were able to collect also blood samples from black rats, so we performed the detection of neutralizing antibodies by a commercial kit. Boringly or fortunately, all animals test negative for SARS coronavirus 2 RNA detection, and black rats test negative for neutralizing antibodies. We were unable to collect blood samples from all the other species. It would be very interesting to control the exposure of these animals, but we couldn't. So from this study, we can conclude that the urban and non-urban wildlife species studied did not play a role as animal reservoirs, at least since now in Catalonia. Until now in Catalonia, and however, the presence of SARS coronavirus 2 in wildlife species in other regions from Spain or in other countries highlights the need to continue monitoring wildlife species to control the establishment of new animal reservoirs of the virus, to prevent the emergence of new SARS-CoV-2 variants that could pose a major risk for global health, and to avoid the threat for the welfare of wildlife. So thank you for your attention, and to my group, and also I would like to thank the Public Health Agency of Barcelona and Torreferrusa Wildlife Recovery Center, who provided the samples of this study.
Okay, thanks very much. Negative results are important, so don't feel bad about only having negative results. I've got lots of negative results. Just in the interest of time, I think unless anyone's got a bursting question to ask, we should move on perhaps.
Actually, you have more data in the poster outside if you
Yeah, so I was going to end by saying thanks to all three of the flash presentation presenters, and if you want to know more about what they were talking about, then please go and have a look at their posters. Thanks very much. Okay, so we will move on now to our keynote by Dr. Zoltan Penzes.
Good morning. Now I'm the only one, I'm the last one now to standing between you and the lunch, I think, so probably maybe about 20-25 minutes more. So first of all, it should not be me standing here, it should be my colleague, Fabian Dornskens, who is you see on the screen, who's the head of our innovation center in Germany, but ironically, he came down with COVID, unfortunately, just 2 days ago, after 3 or 4 vaccinations. So it means there's still something to do, some improvement needed in this case, so he couldn't come. So pandemic preparedness or panic reservation, it is a kind of our choice. So what do we do? What we have learned in the last couple of years? Are we ready for the next COVID-23, 24, 20, 26? Anything we can do better or differently? Or we were just very excited now for 1 or 2 years, and then we will forget what has been done, and we haven't learned too much, and we just be in a panic stage again in the next wave of pandemic. So as a person from a vaccine producer, and also with you as well, I mean, something we can do is how we can improve making vaccines, how we can compress the time to make better vaccines. I mean, all these vaccines were great, but as you've seen, probably many of you, I mean, they were not perfect, so there's lots of room for improvement as well. So they're just going back a bit of time, 40, 50 years ago, this is, I think, one of the first painting by men in a cave in Indonesia, depicting the closeness of humans and animals there, somewhere in Borneo. And that there have been people and animals were living together for thousands of years or 10,000 years. Actually, this situation we have changed a lot, so we are not in caves, except when we are collecting samples from bats, I think, these days. But normally we are out. We have a very complex world today. I mean, it's very interconnected, yet still very, very fractured, so it's a completely different world. Yet still, after those many years and all this technology we have around, I mean, just a very simple virus was able to completely shut down our world today. I want to come back to this very simple virus because coronaviruses have been kind of played down. I mean, 10, 20, 30 years ago, I mean, in 20 years ago, you could fit all the coronaviruses in the world in this room. Today, it's probably not possible. And one of the most pronounced feature of coronaviruses was when most of the coronaviruses were proud of that this was the largest RNA virus known today. I think it's probably still the case. I mean, the largest genome size of 30,000 kb. So it's simple, but let's say we can be a bit better, and we say the most complex RNA virus we know today has shut down the world in most recently. So in SIVA, I myself with SIVA for, I think, about more than 20 years now, since 2003. And then, so the company has changed a lot in the last 20 years, and even including that in 2010, the company has created a vision that as the company was growing, that we want to be a company of One Health. We want to go beyond animal health. And then including, yes, we want to go beyond animal health and to commit it to serve also the animals and see how we can do better with different kinds of vaccines. I think this One Health vision came from, it could be looked at as a forward vision statement that we want to go towards One Health, but it also came within the company as like looking backwards retrospectively a little bit, that we realized that, by the way, in the last few years, we were already a little bit of One Health company. We had a collection of vaccines which was already developed to fight zoonotic diseases. So that also was given us the opportunity to think this further and build more on this collection of vaccines we had in the past. Well, in the past 12 to 13 years, I mean, we have also thought forward this vision of One Health, and all our vision was expanded. And now we are much more passionate. We are very enthusiastic people, including all our researchers around the world in many places we have. And we want to provide innovative solutions for our animals to contribute to the future of our diverse planet. As we've seen, especially now as I'm at the end of the wildlife session, we've seen that there is really a diversity, geography from UK everywhere to Brazil, so different places, and people really looking at all the reservoir of all of these viruses we have around now. I know this is, I did not forget that this is a coronavirus meetings, and myself, I'm also a coronavirologist, so I worked on this in the mid-90s in RNA viruses, or IBV, here in the UK. So, but allow me a bit of time to go to avian influenza to use it as a bit of example, which I won't say that this is the next pandemic, but we have a pandemic of birds ongoing right now. And we can have EU, we can have Schengen, we can have different borders, UK inside or outside the EU. It doesn't matter. I mean, the birds are not asked, and we have a free-flying birds and tons of birds. I mean, around all the world around, and there's no question. I mean, there's no borders for avian flu, and no borders for all these birds to go around in all the world. Well, unfortunately, this is not a picture today. This is not a picture that we can link specifically to a country or a region. Because unfortunately, this picture could be taken probably, I don't say in any country in the world, but in many countries in the world, especially if you're looking at the avian influenza part, and also swine flu is also coming up. It's growing in importance, although that's not as catastrophic, maybe yet, as a few counterpart. I mean, it could be African swine fever, but that's a different subject. We're not talking about that right now. So let's stay with avian flu, which is actually affecting quite a lot of areas. Well, this diagram is, maybe you've seen many of these recently. Unfortunately, I realized that this is already out of date with a very significant change. We just learned a couple of days ago, but let's go step by step. If you see the red dots are everything up to 8 months ago. So up to September 2022, more or less. So we see outbreaks. You've got circles and squares, but the white birds and domestic birds, but let's put them together. So in Europe, everywhere in Europe, we got Southeast Asia heavily infected, I mean, for many years now. And then of the US as well. And there was nothing in Latin America, looks like it, until up to 8 months ago. The green dots here you see is anything from September in the last 6 months, let's say, more or less. Europe everything is everywhere. Let's say in avian influenza, US maybe more than ever. And Latin America, you see, quite interested. Of course, we keep Southeast Asia. And the new thing I learned here as well, talking to Professor Durigon as well, and I also checked on the internet that Brazil is also now infected. So just 2 to 3 days ago, the news, it's only wild birds, but it's only wild birds. You usually got the next step one day, unless you are very lucky. So Brazil is not free anymore, which was the major, I mean, not counting Australia, but which is now the last major area which may have some major economic impact one day. As I understand that Brazil put himself in a state of emergency, I mean, to try to deal with the disease. We have as well a couple of human cases. There's one in the US, I see, as the blue dots, and I think there were a couple of descriptions in the UK, which also emphasizes the link between the avian influenza and the human cases. But I think it's important, especially between us, that we, referring to my title slide, that we don't want to create panic. So the major news of avian influenza is that not all people will die. It's just we have to be aware, and there's a sporadic, and I want to emphasize there's a sporadic spillover, maybe an extreme cases when people are extremely close, exposed to extremely high viral titer, and humans can be infected. But it's not the main point. So birds are the main point at the moment for avian influenza. But how did we get here? I mean, I don't have in a picture, but what was the way of avian influenza getting into everywhere in the world? I mean, it's not in the picture, but probably the most cited, not scientist, but the most cited animal in the world is probably the 1996 Gondon goose. I think you remember. Maybe we should give credit to this bird, where we think everything has started, or where most of the things have started, and then we had several waves of epidemics coming to Europe, and the last one looks like now it's staying for the moment kind of everywhere, coming from Siberia, Northern Europe, the Netherlands, Hungary, Germany, France. They are particularly affected with all the incursions coming in. But there's been also an additional, maybe a new route or additional route from the south, from South Africa, when the migratory route of the birds are disrupted due to climate change, due to elevated temperatures, and people rest, and not the people as well, but the birds rest maybe in certain places where they didn't used to be before, such as in France, and including an area where CEVA also has an R&D center in Andreia, that we have a diagnostic center, so the samples came right to the door. In this case, there's been epidemics in 2020 linked to the spread from South Africa. But how did we get to this stage with avian influenza when we have actually vaccines around? I mean, not even one. We have vaccines of several technology. And this is just a bit of a glimpse of what kind of vaccines CEVA have, and maybe not only CEVA, but we have a range of innovative vaccines, and allow me to go through a little bit on each of these technologies, talk a little bit more. Live attenuated vaccines have been around for many decades, and referring back to coronaviruses, it's been there's a range of vaccines there, which is iBird, iBron, iBros, which is live attenuated vaccines for IBV, a coronavirus, which the animal health industry has actually decades of experience already how to vaccinate against, how to protect against coronaviruses. And at the beginning of COVID-19, I don't believe, and I think I'm probably not the only one, that the animal health experience has not been used enough. I mean, to learn and what the change is and how significant the change is within a virus and how protection would need to be developed, a vaccination would need to be developed against this. So these are specific vaccines for iBird is typically mostly everywhere in the world except the Americas. iBron is a US specific variant, and iBros is a specific vaccine developed for Brazil. So we do need vaccines covering different serotypes, but we do not need a vaccine to cover each serotype, because that becomes impossible from vaccine development point of view. So the inactivated, but unfortunately for COVID-19, I think modified lice for humans were probably out of question anyway, but because they take a long time to develop. But once you have the right safety profile, they are very reliable vaccines to be used everywhere, and still used in billions of doses. Inactivated vaccines, adjuvanted vaccines are classical of course. These are also a good example of coronavirus vaccines. IBV vaccines, typically in this case in Megamoon and Corimune, which have one or two components of one or different serotypes vaccines, and also if the Chinese had asked the animal health industry, then they probably would not have done a killed vaccine for COVID, because I think that's probably was at the bottom of the efficacy of all the different versions which were created, because coronaviruses are typically not very good antigens most of the time. Let's say in inactivated vaccines, they need a primer, as the way it is used, for example, in the avian vaccinology for chickens. There are also swine vaccines in this category, respiratory flu and flu pen, also to combat swine flu around the world. There's a bit of family of immune complex vaccines, which are, in this case, they are restricted strictly to infectious bullseye disease, which they have a combination of live virus plus antibody. which helps to modulate the release of the virus and helps to overcome the monoclonal antibody in certain species. But this kind of solution is really not working for every kind of disease. It's really a specific for Gumboro disease. But the next batch of vaccines, vector vaccines, where of course there has been a great example in a COVID-19 as well. Lots of vector vaccines have been created and designed. This is also has been a great success for the company, and also for, if I stay with birds, as well, that have been turkey herpes virus, fowlpox virus. They've been a very efficient and widely used vectors in the past 20 years, and has completely transformed the industry, or let's say, or the poultry vaccine in the industry. And it's if you notice, you do not even if I do not tell you what it is, you recognize that there is no coronavirus in the vector vaccines. So no IBV, for example, which has been around there. And then despite that there has been 20, 30 years of research of trying to generate a recombinant IBV or vector vaccine, but so far nobody has succeeded. So I don't want to go into the reason why not. But everything else is okay, nearly everything else is okay in the respiratory field, but no IBV, no coronavirus vector vaccine has been done for animals. At this stage, so we got Newcastle, laryngotracheitis, things like that. And with what we do have, avian influenza, different versions. As you see, Vectormon AI is against H5, high pathogenic flu, and Newfoundland MD-H9 is against low pathogenic H9. So those have been around for quite some time, especially Vectormon AI, close to 20 years now, and has been very effective in protecting against high pathogenic flu. We have subunit vaccines, PCV2, and also different things produced in baculovirus or E. coli, different versions. And I think this was the category my colleague from Zoetis mentioned, a description of our COVID-19 vaccine used in the zoos produced in eukaryotic cells. And now, thanks to COVID, or not thanks to COVID, I mean, the animal health industry is also, we are not behind the human vaccine development areas. And we had a program of working on RNA vaccines for quite some years already, even we started before COVID. And now with the COVID, we had an additional boost, let's say, to this technology, and we could speed up, or we had to speed up, and we actually speeded up the development of these kind of vaccines. So we're building on the success of human mRNA vaccines. The difference between human RNA and, let's say, the CIVO, let's say, RNA or animal or RNA vaccine used here, that we're going for a self-replicating RNA instead of just the messenger RNA. This is partly because it's driven by the cost of goods. I mean, you cannot get $10 a dose for a chicken vaccine, or not even for a pig vaccine. So we need to go for scales of economy, which means that we can go 10-fold, 100-fold, much lower in the dose with a self-replicating RNA, and we can have the same effect as with messenger RNAs, with like Moderna, 100 micrograms. Now we are talking about in the microgram range with the RNA vaccines. They, of course, they're very quick to develop. We can have most of they are DIVA, depending on design, humoral cellular immune response, so they can be very, very efficacious. In the CIVA system, we've got a motif in the RNA, which is helping to go around the innate immune response. We've got as well the engine, which is the viral RNA polymerase. It's based on the Venezuelan equine encephalitis vaccine strain, the TC83. And we've got as well, of course, a different gene of interest, which in this case, what I'm giving you an example will be the H5 of H5N8 of the current epidemic strain. And I do have a little bit of data. So once we thought that if we were so quick if avian influenza is around, and then we have such a tool around in the company, how can we use it, and how can we target to be better prepared for a next pandemic? How we can make it something very fast. So we created an experimental vaccine based on this RNA system, using a gene from current epidemics of H5N8 virus as a European gene. And we got a special permit from the Hungarian Ministry of Agriculture to run a field trial in Hungary. And we went for really the hot stuff. I mean that we picked a farm in the area of Hungary, which is every year is hit by avian influenza. And it's an area very dense in ducks and geese and poultry in a watery area. So we selected the farm, a goose breeder flock in this case, with 1,200 animals, and which were divided into two groups. Simple, one group was vaccinated with self-amplifying RNA, expressing an AIH5 gene with a proprietary carrier system. We call it a LION, which is lipid inorganic nanoparticles in this case. So we vaccinated 2 times in September 2022, and we gave a booster, because we need a booster in this case with this category of vaccines in 4 weeks later. And then we let half of the birds unvaccinated. They were like 50 meters apart, I mean the two stables in this case. And then we called for nature to do the rest of the job for us. It was and that everything happened as if you would designed really in a laboratory experiment. Natural challenge came at the peak of surgical response, something when you would design at 6 weeks post second vaccination. And then you can vote what the result is. I don't know who votes that the vaccine worked. Okay, who votes that the vaccine doesn't work? Okay, let's say 2 shots in goose breeders first time, and this is the outcome. What you see there, the red peak is mortality of the non-vaccinated group. This means a challenge came at the end of November, and then within 10 days, these are daily data, and then with daily mortality, with number of birds, and at the peak we had 100 birds dying on each day. So within a 10-day period, 75% of the birds were lost in mortality. I mean, we had a special permit to keep the experiment going in this case. I mean, for data collection purposes. Normally, the flock would be exterminated, of course. So we lost 75% in 10 days, and at the same time, the vaccinated birds, we lost, I think there was 1 or 2 birds which were positive to AI, but there were 1 or 2 mortalities due to miscellaneous reasons, so practically it was close to 0 mortality in the vaccinated birds. Then we looked at, contrary to my colleague in Zoetis, we did look at shedding in this case. So normally the experiment would have been in peace. You know, we would not touch the birds so many times. But when we realized that something was going on, then we quickly had to change the plan, and we took samples every second day from 20 birds from all the different groups. And you see the non-vaccinated birds, we had a titer of tracheal swabs up to 6.2, 6.6. Quite high level of shedding, whereas in the vaccinated birds, the shedding was 2 to 3 logs lower, typically. And also, if you compare the length of the shedding, the vaccinated birds, the non-vaccinated shed maybe over a 2-week period, more or less 2 to 3 week period. And then the vaccinated birds, the shedding was confined really to, let's say, more or less 1 week or maybe a little bit longer, so much less in this case. So it was we could confirm that due to this experiment helped by nature, we had a very good efficacy, and we had a huge reduction of shedding, so much in a month, so much in time for the vaccine. So then back to, are we ready for what do we do next time? What was the history of making vaccines, developing vaccines in last few decades? So back in many years ago, we needed, looks like, several decades to develop vaccines. 42 years, I've got examples in rotavirus, 14 years to reach 40% coverage. COVID, looks like we did learn something over the many years, so we became maybe, let's say, 1 to 2 years, let's say, of coming out with good vaccines. Best practice is stated as 250 days, but the target is now, including according to a report published by CEPI, is now to be ready with a vaccine of within 100 days. But the question is, what do we mean? What do we mean by ready with a vaccine within 100 days? So let's see a little bit of schedule. What the 100 days is, and what we can do within 100 days. Going back to this example of RNA vaccine, this H5 vaccine, to see when we saw the epidemic coming, then we decided we had an internal go with the company in December 2020 to go for an H5 vaccine for let's say, for ducks and geese in this case. And within 100 days, in May, we synthesized the gene. We've done all the cloning. We've done all the production of vaccine in small scale, in laboratory scale. We started an animal experiment, and we had the challenge results and everything. We had everything that the vaccine was working in May. So let's say this is roughly 100 days or maybe slightly more. So within 100 days, technically it's feasible to design, to make, to test in R&D scale, and to run an experiment and see the results. So, but is it enough or not, or where are we on the rest of the path? Well, the rest of the path is not 100 days. The rest of the path is, I don't say 1,000 days, but it's definitely several hundred days. As we look further, we were in close contact with the French government, the CGIAR, and then we have learned that there is a government interest to work on a development of an experimental vaccine, because it's non-sustainable to kill millions of birds every year and wipe out part of the economy. And this is especially referring to, for example, the waterfowl in France and also in Hungary, which is one of the most affected areas. So then the French government announced that they are ready to test some vaccines in October of different suppliers. We were one of the applicants, and they did run the testing in May 22. And I think the report is now publicly available on the French government website. Somewhere I've seen the email a couple of days ago now. And now we are, and then in the meantime, we're working on a regulatory package. And the regulatory package actually has been submitted to the French government for exceptional use. I mean, Europe is a bit more difficult than US. Looks like in US you could get a special permit and emergency vaccination. Europe looks like there is more thinking about this, how we will do all the politics and all the commercials and export-import rights linked to avian influenza. So 100 days is possible technically. The rest of it is not really up to us. And also industrial scale-up. I mean, if you have a technology which is already existing within a company, then quick scale-up is possible. So if you already established RNA production large scale, first time, of course, it's not 100 days, but second time and third time, it can be around 100 days to produce, especially if there's a footprint. RNA production is quite small. So 100 days, we can do 100 days in R&D in a small scale, but the rest of it is really not so much. Is really not so much up to us. So we need you, all of you, your friends, your colleagues, your country representatives. I mean, to try to be more flexible and open doors, remove these roadblocks. I mean, at the same time, we have to maintain, of course, that we are scientifically correct, and we come with safety first, of course, but then the second is efficacy. This needs to be maintained. So we're back to One Health again now. I mean, especially now, as I say, at the end of the wildlife session, I mean, One Health is not only 28%, which is the human side, so we got a much bigger stake on the animal side, and as we see, we are seeing viruses being discovered. Where everybody who looks like is finding a new virus, a new family, new genera. So there's a lot more to do, and all of these 72% we don't know. I don't say we don't know anything, but we don't know enough. So that needs to focus on that one. So in this world, SIVA is active. So we are here. That's one reason why we are here. Why we joined the consortium. So we'll be with you. We're working with not with everybody, but with some of you, collaborating how we can take it forward, how we can contribute to improve animal health in the wildlife as well, and then in such a way, improve the safety of humans and have less diseases. One example is where we are very active is rabies vaccination. Again. In different species around the world. I mean, within the company, we've got, I think, 3 different technologies for rabies vaccine. We've got live attenuated, we've got vector vaccine, we've got killed vaccine. So everything is suitable for a purpose. You need to find what is the best technology and what's the best route of vaccination, because having a vaccine is one thing, to applying, but it's you go nowhere with a good vaccine unless you have a very good route of application and unless it's applied properly. We'll discuss this more in the workshop. I think the routes of application would be one of the questions where we can talk. And then one more thing, this is the koala here is not only for illustration. The koala serves a real purpose on this picture. It's meaning that just in a few months ago, CIVA established a wildlife research fund, which is a legally independent entity. And somehow we want to handle separately all those requests, questions, and issues around the world, which somehow do not fit in the commercial setting, because commercially you cannot make a vaccine for koalas because it would never survive. So we need a different setting, and to working with you, with some of you, I mean, how it can be done. And indeed, koalas was maybe the catalyzer for CIVA to establish this wildlife research fund. There's nobody here from Australia, no? Oh, you. So that's because they have a disease affecting reproduction, and the population went down a lot, and we kept receiving lots of requests. I mean, if we can do something, and because we have a vaccine which could be used to protect koalas, I think now this is on the way of working on it. And so koalas may be the first one. We do rabies, or maybe we can do bats somewhere maybe in Brazil, where it's an issue. So there's many things to do, especially if you talk to some of you in the breaks. There's a real need for this kind of activity. And I think with this one, we are the only one in the animal health business to come with this kind of initiative. Thanks also to Martin and to my colleagues who are not here. So thank you again. So panic preparedness, or are we ready, or are we going to learn this one? So let's hope we'll learn. So we turn the scream into a smile, and let's be ready and come with innovative solutions. And I'm really happy to be here with you, and it was great to see and talk to many of you in this meeting, in the breaks and during the dinner. I think we should have had a night in a pub as well somewhere. Maybe that would have been even better. Contribution, but I think we have to work together and take this opportunity to move forward and work together to keep this smile and be ready at the next time when something will come, because I'm sure something will always come in this case. So thank you very much, and now I think the door is open for lunch, unless you have some questions.
Thanks very much.